Well, I want to thank the organization, obviously Nature, and also NASA for inviting me to do this, because you get, there's all these the good old days and stuff like that. But I found out as you get older, there were older days, but there were only a few of them that were really, really good. Okay, and that's the ones you remember, thankfully. So I think we're kind of selected in this environment to actually be that way. So I chose this because I thought that half of this would be pretty much straightforward, and the other half will probably get me in trouble. So, which I'm used to also. So do I control these or you control them? Like I said, there's a clicker on the podium here. Okay. And push the right button, let's see. Push the wrong button. Forward. There you go. All right. And I'm just going to give you a little bit of this. And some of this you've had already. They talked about it on the first day. But I probably will take a few of those concepts and embellish them a bit more. There's also the idea here that a lot of what you're interested in microbiology is applied microbiology with regard to crew health and those kinds of things and the environment that they're in. So basic metabolic processes that might be important to human health and operations during spaceflight can evolve from this, okay? And we can even get more once we get into planetary habitats. And you're all busy right now with the proteomics, metabolomics, genomics, and obviously the microbiome itself. And there's a whole series of others that could get co-opted into this. The applications, I have some things to make about statements for applications. There was a lot of talk about bioregenerative systems that we could actually bring to space and help use them to really put a lot of resources into a cyclic type pathway. And I think there's real value in that. It's not that I'm trying to dissuade you from it. But I think that we can have a short discussion a little bit about what some of the foibles of that might be, given that the environment that we're in. And obviously the pathogens, you've had a lot of discussion on, I won't. But I think that it's still important to remain vigilant about the environment. And I know there was a lot of discussion about the environment, and I was excited to hear that they were concerned about the environment with regard to how it might change. And I think some of the things that we can talk about a little bit later in this might help us on how it might change and how we might manage it. The myome you already dealt with. I think that the last thing that's on here sounds a little bit far out there, I didn't smoke my lunch or anything when I wrote that. Because observing evolution is something that you can do in microbes so much better than we can in other types of living systems. And I'll get to that too, because I think that's an opportunity that you can't pass up. Because there's an excitement level to the science community and to the quasi-science community that's out there interested, okay, of what could happen under those circumstances, what we could observe. And so I think that not only for the sake of the explorers, the habitats, the vehicles, all of those things could be impacted by the kind of findings that you could have there. The challenges. Now, these are only some of the challenges, and they have a lot of the same challenges you have in ground-based research if you're an academic scientist and whatever. And I spent my first 20 years doing that, and then the next 26 years doing NASA. So I've been around a while, in case you know. And so I'll try to not wax too much of that. Okay, but I think the challenge is experimental design. Just in the discussions that I heard today and some of the ones that I heard yesterday, experimental design becomes critically important. Okay, and design in the context of not only what Rob was talking about, as far as the statistical platforms you might use, and how you might change the way you think about the results that you get from those. But I think that the design also, in refining the question you're trying to answer, and this I did with graduate students for years. They'd bring me an experiment, Dr. Pellis, I want to do this experiment, and I look at it, and it's a shovel full of data, and that's about it. And I said, what question are you going to ask? What's the question? And do you have the question refined to a binary type answer? A yes, no. P less than 0.5. Hopefully not. Okay, it's sure that the goal is obtaining answers, and success is not measured by the amount of data you get. Now, I'm a geezer, and that's why I'm saying that, because when I got to NASA, the first thing I did was meet with all the people that I was going to be working with and supervising. And I asked the question about the science. And they said, oh, we have very successful science. I said, based on what? And they said, well, we collected a lot of data. Okay? So they got this avalanche of data. And I said, but what answers did you provide? And the people who were actually conducting the experiments couldn't provide that. They couldn't give me the answers. And so it made me focus back on it and say, don't get walked back into this again, and get your success estimated based on the amount of data that you get. Managing interpretation of the results in the context of good experimental design principles. And I can show you. In fact, I'll tell you, because I'll just preempt those slides. Early on, we found out that cells which we theoretically believed would not survive in space because of becoming diffusion limited. All right? And we'll explain that if anybody doesn't know. I know the scientists know it, but there's others here who might not. And it turns out, by my estimation, based on the controls and everything that we had, we actually observed cells adapting to mass transfer delivered by Marangoni forces. That's surface tension-driven convection. And it's only apparent, really apparent, in a condition where gravity is diminished substantially. So it was one of those things that, not having the right control, we could have been misled in that answer. Avoid overstatement of significance. This is another task that was 2 diseases for sure that I can recall, because there were slides with those on it. We were curing cancer, okay? And it was pointing, the arrow was pointing to a facility, space station facility, and it says cure for cancer right there. And the other one was diabetes. And in the earlier phases, pre-ISS, there was a lot of work done in the propagation of cells in space and the separation and purification in space. The idea here was that they were going to make the perfect preparations of pancreatic beta cells that they could use generically in patients with type 1 diabetes, okay, by giving them a beta cell transplant. And, well, it's beyond imagination. I don't know who was imagining it, because you had to have some kind of mind-altering whatever to actually put it down on paper as being such. So we got ourselves in a lot of problems. We got a little sideways with the NIH. We got sideways with a couple other agencies in doing those things. So in our exuberance to actually take our own advocacy, bring it forward, and to be able to get the kind of funding and things we need, we have to stay kind of inside that fence where we're not overstating what the possibilities are or overstating what our results are saying. And then this last one here I'll deal with in regular detail, and that's establishing the response curve for critical biological processes, okay, in a fractional gravity setting. Now, Howard Ross, who was chief, he actually started out as a combustion physicist at Lewis, what it was called then. Now it's called Glenn. But he started out as a combustion physicist, and he ended up being chief scientist for NASA. Yeah, an OVPR, excuse me. That's right. Office of Biological and Physical Research is what that is. All right, I had him, I don't know, chief scientist came to mind. Anyway, he came up to me one time after a presentation that I had done, and he said, you know, he said 90% of your problem with growing things in space, cells, bacteria, and this kind of stuff, is actually a fluid physics problem. All right, so I had to start listening because I didn't know enough to make any kind of an answer that would sound halfway intelligent. And hopefully by the time we're done, I think you'll see why I say that. Okay, some of the quotient, things that we can use microbes for in space is what's the potential basis of life response to microgravity. Now, this is a basic organism. It's a fundamental, low phylogeny type organism, but has enormous conservations of a number of processes, okay, all the way up through to human beings. And so why not take advantage of that? And we'd like to find out what are intrinsic responses. In other words, does this organism have a gravimeter? We know that there are certain plant type cells, root hair cells, have a gravimeter in them. But do these have gravimeters? Do my liver cells have gravimeters in them? Is there something that's measuring the system with regard to its force of gravity that's exerted on it? So that would be a direct effect. And I can tell you right now, I stood up in front of the National Academy of Sciences in 1995, somewhere around there, 96, and said it's highly unlikely that there is a gravimeter in a living system. Much of what we're looking at is after Howard Ross got a hold of it. Much of this is actually fluid changes in the environment. And the microbial response to environmental changes became very important because microgravity enormously affects the environment that we're in. And I know on the first day they talked about density-driven convection and phase separations that don't occur in space. So, when you mix oil and vinegar together in space, you don't get the same partitioning that you would on the ground because more dense things don't sink to the bottom. So that's a really critical type of observation to not just retain, but to accommodate as we design these experiments and as we try to ask the questions in space. Now, all of this talk is about gravity. And they're basically these four types of interactions, physical interactions in nature. Gravitational is one of them, electromagnetics, strong and weak submolecular forces. But interestingly, when you look at this, gravity is actually the weakest. Okay? The amount of energy it requires to build a covalent bond or to break a covalent bond is far in excess of anything I would do in tearing this piece of paper. And so you can see now when you look in the context of that, if I want to oppose gravity, right? I think I just did, okay? You can oppose gravity. We can propel things into space, propel things up into the sky. So that's something to keep in mind. And the intermolecular interactions that are occurring in space occur with the same tenacity that they occur here. So whether we're looking at covalent bonds, non-covalent bonds, other types of molecular interactions that take formations of macromolecular assembly, all of that stuff can occur pretty much the same way as it does on the ground. There's some exceptions to it. All right, these are the things that you got as a way of characteristics in the space environment. Most of it is based on fluid, but the decrease in hydrodynamic shear, a lot of the culture work that we do in microbes is done under stirred conditions. Okay, and in that setting, you get two types of shear forces. You get mechanical impact shear forces, and you get hydrodynamic shear forces. That is, the water moving past the cell. And, if you have trouble relating to that, if you're riding in a canoe down the river, and you're just going along and everything's fine, There's no shear on practically no shear at all on your canoe. As soon as you hang up on a rock, you'll find out what shear is and how much damage it can do to you. And so shear is something that can be a benefit to living systems. In other situations, it can be deleterious. The type of, and I mentioned this before, the type of convections that can occur under microgravity conditions are surface tension-driven convections. These are called Marangoni-type forces. And they're considerably more subtle, thousandfold more subtle than the shear components that, the kind of convection that you can get out of density-driven. This results in a decrease in mass transfer. No sedimentation. No sedimentation we normally think of when we're talking about cells. It changes the way we have to handle them. We're so used to, if we want them somewhere, we may slam them up against some wall where they can hold them, the wall of a test tube or whatever. They'll sediment. But that doesn't occur in space. And so you'd say that's probably a disadvantage, and it can be for mass transfer. But it could be an advantage for other things. Believe it or not, cells in space have a propensity to assemble when suspended for long periods of time. They will actually come back together. And Don Pettit, who's an astronaut, he and I had a lot of discussions about this, and I told him, no, this is what happens. This is how we get tissue morphogenesis in space. We take mammalian cells and put them in a microgravity setting, and they assemble and they start coming back together, and they morph back into the tissue they came from. A very interesting phenomenon. So he was on I think he was on, he was in space station we did it. He took a Ziploc bag and filled it up with non-dairy creamer. Not filled it. He put a couple of tablespoons of non-dairy creamer and shook the hell out of it. And then just hung it up there and let it stay there. So the non-dairy creamer's sitting there. He was expecting that he's going to see the subtle gravitational accelerations that are occurring on the space station as it goes around the Earth. And it would partition to one side. That was his idea. And in fact, it didn't. What happened is the particles agglomerated in there. Okay? They were in the bag in there. Same thing happened with, he's a coffee drinker, so you can't help him. Instant coffee, he did it with that. And then he took mica flakes. They had some mica flakes up there, and you usually see that in your potting soil. The little things that are shiny is the potting soil. But he took those and he suspended them in water in the bag. Took longer, because it takes longer for them to move through the more viscous fluid to get to each other. But they did the same thing and agglomerated. So this is an observation that together, physics and us talking, we began to understand some things. And he said, this is amazing, because when we look at this, we had no idea how dust gets to become rocks. We know how rocks become planets, but we don't know how dust becomes rock. And so that was his big gig with the bag and the non-dairy creamer in it, is that that was a critical step. That's how you get from particles to rocks. So it can help us to keep thinking in these contexts. I think the same thing is probably going to be true when you look at microbes. You look more and more as to how they build the basic structure or the initiating structure for biofilm. And I think that this probably won't go away for quite a while yet. All right, this is explaining that. It has to do with the pressure gradient. I'm going to get rid of that. I just want you to know that when we get a situation where there isn't any density-driven convection, then basically the cells are acquiring nutrients from the media based on fixed law of diffusion. So it's a much, much slower process. It's a much slower acquisition of the nutrients. And it's also considerably less in a way of the dissipation of waste products produced by the organism itself. So big changes that can occur, and it's all in the loss of this critical feature of diffusion, of convection. All right, you've already seen this, I don't need to do that. They didn't die, and I don't know for sure exactly why they didn't die, because their mass transfer is not consistent with what it would be on the ground. But we theorize, and most of the physicists would tell us that these cells would eat themselves into a zone of depletion and starve to death. Or they'll excrete themselves into a zone of toxicity, right, by not being able to get rid of it. But when we look at this depletion, O2 is the first thing to go, and we know that glucose is not far after it. So at some point in time, there's probably going to be a sufficient depletion to get it to make them die. So you can see microgravity is a unique environment and has substantial impact on these processes. And these are just a few that we're talking about. The research in microbes is so critically important, okay, to be able to set what the basis is for a number of these process changes. We can look at this metabolically, we can look at it morphologically, we can look at it, and you just add up the ways. And once again, you're dealing with microbes, you're not dealing with finicky, liver cells or something like that. Well, I guess I'm done. Oh yeah, I am. Look at that. It went the whole way to the end. Get rid of these. All right, it is unique. Yeah, we got that. This is an interesting concept. This came about with me one night. It's a long time ago, 30 years, something like that. It was the middle of the night. I woke up, and a movie was on, and it had Charlie Chaplin and Paulette Goddard in it, and it was about Hitler. It was the great dictator or something like. I don't know what it was like that. But Charlie Chaplin was sitting there watching, and they were talking and get back. He got up and he went over to this big cradled globe. The Earth. He picked it up and took it over, and he kicked it off the back of his heels, bounced it up there. And he's doing this back and forth. And I was watching, and when I watched it, it's not changing orientation. He'd kick it up there, and it would just go up, then it would come back down. You would expect it to spin, and it didn't. And it was wired, so to speak, not connected wired. It was wired because he had an asymmetric distribution of mass inside the cell. And so we wonder why certain cells orient the way they do in given settings, cell culture type thing. And it's probably because they have an asymmetric distribution in mass. So in any event, you never know where you're going to learn something. You never know where I had to write it down. Of course, it was practically illegible the next morning, but it didn't matter. So as life evolved on this planet, there were a multiplicity of changes that were invoked in it by the chemical environment, the physical environment, all these various things. Okay? For many of these, there are clear examples of the role of changing physical forces in evolution. Amongst the most notable of this is gravity, because gravity has not changed. Remember, changes in these things are causing it. Gravity has not changed. You go look at textbooks in biology and look for anything in there that's got a formula in it about something and see if you can find a G in it. Now, today you might find it. But as of about 10 years ago, I made a bet with a student. I'll take it on a dinner at an expensive restaurant here in Houston called Tony's. He lost, okay, because he couldn't find one. But now I think you're probably getting closer. But the reason why is it has been constant for 4.8 billion years. and is treated everywhere in non-physics as a constant. We don't even think about it. The other thing about it is it's becoming more and more apparent, okay, that there's little or no organized genetic memory or a suite, okay, for life responses in low gravity. There are mixed evidences of suites of different sizes, shapes, and all that stuff for different things, but there's no unifying suite for that change. So that just bespeaks the fact that most of the life that's on this planet has never had to deal with this, has never had to deal with hypogravity. And so that's what, and if it did and evolved, it's lost because that particular characteristic was not retained. Which brings the importance of understanding the gravity. It's not apparent to everyone how gravity works. And when you talk to people about this, you have to explain to them that the only way you get gravity, low gravity, is in space. We have analogs and systems here on the ground. They can mimic some of the characteristics that go on in living systems, but it's not a simulation of microgravity. And I started that a long time ago, and it's still in the literature now in that way. But if you're looking here, this is G on this axis, and that's your favorite biological response. Pick the one you like to do. Growth of microbes, something a microbe is going to do, whatever. And we have data here, and Craig Kudrot said this. He just kind of was picturing this graph, right, when he said this. I got these data here, and I got this data, out here at 0.1 of a 0 G, and that's the way it looks. Okay, now draw the line, because we don't have the data in the middle. There's precious few of anything that's been done so far, and we have asked for this since 1998. Not a very difficult thing to do. The hardware is already made and everything. It's small. You could do it. So what would you get? What would you guess for, some kind of a response curve? Power curve like this, an exponential curve like that, straight line, sigmoid. What would you think? This is a biological response. Think hormone receptor interaction. Think of any of those kinds of things you had in biochemistry class. They're sigmoid. Interesting thing about sigmoid relationships in biology is most of them occur within 1 to 1.5 logs of normal. The yellow is not the curve. My engineer friends wanted it to be this curve because it says at 10 to the minus 3, you got enough G, you should be able to do everything you want to do. Right? Well, it's not. It's more likely going to be this. It's never been done, so this is strictly hypothetical. And, I deserve all the ass kicking anybody wants to do if I'm wrong, but for right now, this is what I think the hypothesis should be. I don't know if we'll do it with P less than 0.05, but it should be the hypothesis. And so to me, it's so important because it actually dictates a whole bunch of things, not the least of which could be the design of habitats, the design of processes that we're going to do in space with regard to maintaining life. Knowing what the thresholds are. Does every tissue in the body, cells from that, have the same threshold for function? Don't know. Will your immune system be the first to go? Okay. Will your kidneys be the first to go? Neither one of them are a good day. So I think that this is, when I say opportunity, this is an opportunity. Right? And it has a straightforward answer to it. And I took this and I took only the part of the curve that I wanted, and I put in the moon, Mars, and Earth on it. And the only reason I put it on there is that when we go back to the moon, and when we're going to Mars, we'd be fools not to take the opportunity to do the kind of experiments that we need to do in those actual G-force environments without any artificiality to them. I think the other thing about it is, in the beginning, we can do bacteria, we can do cells, we can do small packages and that kind of thing. But you also have the opportunity in those two environments to put the dots on the curve for mammals, mice, for insects, for other things, and get a much better idea, excuse me, of who response is. Now, keep in mind, I'm saying this like it's almost canonical. It's only that way because I've been thinking about it so long, I don't have a shred of data to prove anything that I've said. And honestly, like I said, I'll be the first one to do the kicking on myself. But at this time, this is the most compelling. And I think that the value of this to the space program is twofold. There's a tremendous value, operational and design, that is attached to this. There's a tremendous value scientifically to the basic science community, because this is something they won't have an opportunity to do ahead of us unless they pay somebody, to do it. And it could happen, but I'm not sure there's anybody thirsting for this knowledge like I am. So, could be. The things you've already talked about, all these, but when we look at this is kind of an interesting portrayal here. Obviously, it's a bacteria on the ground. You can see it doesn't look any different in space. It comes back and it doesn't look any different than it did before. And if this were a mammalian cell and didn't have a rigid cell wall and a pliant membrane, it would look substantially different. So the mechanisms that are going to occur in these kind of cells are going to be vastly different, in many ways than it would be for a mammalian cell. Because a mammalian cell would have this general pleomorphic form. It'll become, it minimizes its surface area to volume ratio. So it becomes spherical, and while it's in space, it comes back, it's reshaped again, and everything's fine. Gene expression changes, which you all know about. Shift to secondary metabolism. I don't know if you had much talk about that at all. But in the early days of space microbiology, National Academy member Arnold Domain spent a lot of time cogitating about this and doing experiments and models to actually explore this. The fact that they shift to secondary metabolism. They go to gluconeogenesis, basically. And they secrete a lot of things. And they change in the way they cooperate with each other. And this came up before when we were talking about the bioregenerative systems, and we talked about mixed systems. We're looking at mixed populations that are in those. There is no a priori guarantee that you've got a sewage disposal plant that works here on the ground that will ever come close to working in 1/3 G, given the same organisms. And I was asked, Dan Golden got us hooked into an American Society for Microbiology meeting years and years ago. And we had a panel and we talked about stuff and everything. And people were really very nice. They had a lot of good questions. Arnold Domain put up his hand and he asked me, he said, Neal, he said, if you had your choice today, what experiment would you do in space? I said, I'd make sauerkraut. And he looked at me for a moment and he kind of smiled, and then he realized what it was. That's a sequential fermentation. It starts out with Leuconostoc mesenteroides and ends up with lactobacillus. Leuconostoc sets up the pH so that the lactobacillus will grow, and it gives you the ferment that you get. So I could actually visualize a mixed population in a setting where it was fairly refined. It's not like a sewage disposal plant. It's got 2,000 different organisms in it to be functional. You only have to have 2 here, and will they cooperate? It would be a very interesting experiment to do. I'm not sure the crew would appreciate the smell, but hey, when the restroom's not working, they actually know what it's like. So, novel antibiotic resistance and quorum sensing. Quorum sensing is the way organisms keep their populations in somewhat balance. They're not balanced like this. You've got all the populations balanced. You know, some are here, some are there, but it's evanescent. And so it goes up and down, and it's maintained. It maintains a reasonably functional status in doing that. But when we go to microgravity, we don't know if it's going to be that way. I think we can do some simple experiments right away, not doing anything really complex. You don't need to send a sewage disposal plant up, but right away to tell you whether or not it's going to be sustained. If it maybe might be sustainable, you could do all the work you want to do to make one of those here. Then it doesn't guarantee that you're going to get the results you want. There are some changes in virulence that now have become almost canonical. I think several organisms now, it's clear that they do. And we'll probably continue to face more being added to that list. And the interesting thing here is, obviously, there's some kind of a mechanosensitive response that's going on. In bacteria, right now, my small pea brain, super soft biology, not the hard shell biology like the microbiology. The microorganisms are, tell me that it's probably, I'm guessing, but probably mechanosensitive channels, because they're conserved phylogenetically from protista all the way up to primates. So it could likely be that that's what it is for this. It's not like it is in mammalian cells. Mammalian cells, it has the mechanosensitive channels, but it also has primary cilia. Okay, and I don't know how many people have ever even heard of primary cilia. Every nucleated cell in the body at some time in its development has a primary cilium. The primary cilium is not motile. It has no pair of microtubules down the center, none. Right? So it can't move the cell along at all. All it is a very exquisitely designed force transducer. That force transducer can measure forces in the femtonewton range, 10 to the minus 15. That is incredibly subtle flow, okay, that would be around. And so I think that we're looking at those, and we're going to see a difference in the sensitivity of these organisms versus mammalian cells when it comes to responding to external forces. All right, we're getting out of here pretty quick here. Okay, one high impact opportunity that I'd like to talk about, okay, is looking at things that are happening in space and the kinds of cells that we're using. And if you look at the number of generations in an ISS increment, oh boy, okay, in an ISS increment, okay, obviously humans, 0.02, okay, generations, but mice, we get 2.5 to 3, somewhere around there, generations, in an increment. We could probably study some things. I'm not sure how well we would do in chronicling heritable differences. But when we get the mammalian cells, between 1 and 2 divisions a day, that means on a Mars mission, your gastrointestinal epithelium from north to south will turn over 1,000 times. Okay? It's going to turn over 500 to 1,000 times. And the big question is, what is the fidelity of that replication? What are the possibilities that we have errant events occurring that are heritable and retained? We don't know. Bacteria allow us to get there a lot sooner. Okay? Many thousands of generations in an increment, in a 180-day increment. So, looking at that, like I told a couple of crew that were at this meeting, he said, well, who cares why? I said, you could have a grandstand seat and watch evolution take place because of the short generation time. And as a scientific opportunity, I consider it monumental. It's not trivial. It's something that could literally shake a lot of brains throughout the country to start thinking about these things. And so I'm with you. I think bacteria are the way to go. And that's how I started out. I worked on Microbacteria and Listeria monocytogenes. All right. Challenges. Right now, if we want to be able to do this, we have to look at how we're going to attain the number of microbial generations during long-term growth spaceflight. And it's difficult due to sometimes practical consideration and sometimes hardware limitations too. But I think that it's just like when we told Mike Suffordini at Space Station that we need to have the following and the following and following things on Space Station to be able to do life science research. And he said, well, and I said, look, if you've got it, I can produce people who will want to use it. And I said, be ready, because the line's going to be out the door, down the hall, out of the building, and around the block. And he said, well, there's no way we can accommodate that. That doesn't mean you shouldn't advertise that you have the capability to do it. That's how you can be sure that you get the best of the best out of it. That's what we do in science already today. So I think we have to do the same thing again. And this time, we have to be able to, to be able to accommodate this kind of an experiment. And it's in progress here with Evolves. That's a current experiment on salmonella in a liquid medium. And they're going to target getting 300 generations. Now, 300 generations, I think you're going to see mutations. You're going to see some changes. I don't know much heritable and how much competitive there is, because we don't know. Okay, we just don't know what all the selective pressures are, and and least of which do we know that microgravity itself is a selective pressure. Which, if it is very, very profound for how we begin to envision terrestrial life moving to these new environments, most of which are lower gravity. So I think that, and when we look at the challenges, I think the challenges may look a little daunting, but to tell you the truth, it's incredibly exciting. And I think it's a great opportunity, and if it were really, really expensive, like trying to take primates to space or something, it would be, there's no comparison when it comes to the resources that are necessary. Okay, there's no current flight hardware that can do this right now to this extent, to the extent that we were talking about before. I think they'll do it with Evolves, but I'd be very, very interested in seeing what the results turn out to be. All right. I don't know if I want to go through this or not. Getting the theoretical number of microbial generations during a long-term growth in space is often difficult due to practical consideration. And so I should have said that right at the beginning. I don't know what it's doing. Is that at the end? Okay, there you go. The overall challenge is adaptive responses versus genotypic and phenotypic responses. Genotypic, I have a fair idea. Given the radiation environment that there are going to be genotypic changes. What will be the severity of those changes? I have no idea. What is the sustainability of some of those changes? I don't know. The adaptive changes we know very easily. You come back and they're all gone. Okay? Back to normal. You're back to ground state. So that one, I think, is critically important. And then what else in that environment invokes the selective pressure? On both dividing and even dormant cells, for that matter. And see what kind of can happen there. How much G is required to maintain normal? That's the chart that I showed you before. Right now, what do you think the answer is? If you had to look at the chart again, what do you think the answer would be? I'd say by the time we hit the moon, we're bottoming out. We're asymptotic at function, you know. So it would be not that different than 0 G. So it's going to be between 10 to the minus 1 and 0 is where it's going to be. That's where the threshold is. Without a threshold back here at 10 to the minus 3, like some of our engineering consults were getting excited about, without that, that's going to make the design of things considerably different. I already talked about this too. I don't think I need to talk about that again. I just really get myself in a problem. Understanding and communicating the omics avalanche. Okay, for those of you who are doing this kind of stuff, working on microbiome, do you find it when you try to talk about this outside of anywhere, like if like a reporter talks to you? You try to explain what's going on, or the omics data that you've got. You've got the genomics profile for it, and you do these profiles. And somebody over here mentioned it earlier. I don't remember who, but you do the profiles on these. You can't do a snapshot on the ground and a snapshot at 21 days, okay? And look at the differences. Because if you do a sequential study, just looking at one of them, you'll find out that there are different patterns depending on which gene it is you're looking at. Some of them will be constitutively elevated, some will be constitutively repressed. Others will have evanescent, once again, in and out type expressions that go on. Some may go up and just stay consistent for the rest of the time. I don't know. But when you do that, you change a whole lot of the dynamics of the population. Okay? Whether it's a mixed population or if it's a pure population, you're going to change the dynamic. And so it could have some significant effects on the environment, on crew health, and those kind of things. All right, I'm going to finish here because I think this is one of the most exciting times. And I want to be there because it's just too damn exciting to miss the show. The show is going to be terrific. And particularly, I know the pictures are going to be great and all of that stuff, but the science is going to be exhilarating because it's going to be unparalleled with anything else that's been done in history, in science. Right? So, and it's the first step out there. And it's going to tell us a lot about what we need to do if we want to go out there and stay there. On ISS and the way this is designed and the way it's been worked out, it is a very, very good first step to getting ourselves positioned to be able to develop the ways to use this and having campaigns of research that can actually give us bodies of information that have panels of answers in them rather than just more data. Okay? Lastly, and I can't say this enough because my thinking changed after being a professor in the medical school for 21 years. I got here and I started to have to think differently. Talked to a lot of scientists, but talked to a lot of physicists, and talked to a lot of people about how to do things. And this is what makes it very important. Biology, physics, computational modeling, which I have great respect for. Check for for the record, check the prediction of the number of hurricanes we're going to have. Okay? I mean, in my, where I live, my little neighborhood back in Pennsylvania, it worked like air brakes and a jackass. Okay? It's just non-fit. Because it's just that you don't have enough years around for us to actually validate the model. And so, anyway, taking care of big data, I think there's a great opportunity. I'd like to ask these guys who work big data and microbiomes and all this kind of thing, are we making improvements in how we get the answers that we want from that data? And if we are, I'm encouraged by it. Otherwise, I think of that old story about the kid went to visit his grandfather at the farm because his mother said, you need to spend a summer there because of the pain. Right? So the kid's there, and he's working and all that stuff. And the old man comes out, it's about 6:30 in the morning, and the kid is in this massive manure pile his shovel, and he's just working like furiously. He said, "What are you doing?" He said, "Grandpa," he said, "with all this poop, there's got to be a pony in here somewhere." And so here we are. We got this huge, massive data, and it took a lot of effort, and it took a lot of smart people to get the technologies put together to do it. And I think it's still going to take more smarts in the development. I'm looking right at Rob Platt-Snyder to actually be able to get the message that's in there. Where's the answer that's in that data? And so with that, I'm going to stop, and I have to say my deepest appreciation and thanks to you for tolerating this